Hi everyone, and welcome to our module on microtubule inhibitors. Let's start by reviewing the structure and function of a microtubule. A microtubule is a structural element of a cell, and microtubules are polymers which are made up of repeating protein units called alpha and beta tubulin. The structure of a microtubule is shown on the screen here. You can see that it's a long hollow structure made up of repeating units shown in this drawing as red and brown units. The red units are alpha tubulin, the brown units are beta tubulin, and they alternate to make this long arm that is a microtubule. And microtubules are found inside of cells, and they can grow or collapse, and they're used in structures like flagella and cilia. They're also very important for cellular transport to move things around a cell. This is especially important in axons. Recall that the axons of neurons are very long structures, and they heavily depend on microtubules. And if you remember that, that will help you remember some of the side effects of the microtubule inhibitors we're going to talk about in this video. Microtubules are also very important for the process of mitosis. Mitosis is part of the cell cycle where chromosomes are separated for cellular division. This depends heavily on microtubules because a structure known as the mitotic spindle must develop. This is a drawing of the mitotic spindle shown on the screen here, and microtubules attach to the chromosomes and pull them apart so that one chromosome goes to each of the daughter cells that are being formed. The process of mitosis is followed by the process of cytokinesis, where the cell divides. In order to understand how the microtubule inhibitor drugs work, we need to review two important phases of mitosis. The first one is called metaphase. In metaphase, the chromosomes of a dividing cell line up on what's known as the metaphase plate. This is a drawing of a cell with its chromosomes lined up on the metaphase plate. In order for this phase of mitosis to proceed normally, microtubules are very important. They must put these chromosomes on the metaphase plate. Metaphase is then followed by anaphase, where the chromosomes separate and are pulled to opposite ends of the cell, which will later divide into two daughter cells. This phase is also heavily dependent on microtubule function so that it can pull these chromosomes away from the metaphase plate and into the opposite ends of the dividing cell. Drugs that inhibit microtubule function are cell cycle specific. They function only in the M phase. That is the mitosis phase. That is the phase of the cell cycle where cells are dividing, and that's where the microtubule inhibitors exert their effects. We'll talk about two classes of microtubule inhibitors. The first ones are the taxols. The second group are the vinca alkaloids. First, a bit of vocabulary. Alkaloids are naturally occurring substances that are nitrogen-containing bases. They are usually found in nature and derived from plants or trees. And there are many examples of alkaloids which can exert effects on the human body. For example, nicotine is derived from the tobacco plant. It is a nitrogen-containing base. It is an example of an alkaloid. Morphine is also a nitrogen-containing base. It comes from the opium plant, and it is another example of an alkaloid. There are two Taxol drugs used as chemotherapy for malignancy. They are called paclitaxel and docetaxel. Both of these are alkaloids derived from yew trees. They are mitotic spindle poisons. They bind to microtubules, and they actually enhance tubulin polymerization. It's very important that you remember this. They don't inhibit tubulin polymerization, they enhance the polymerization, and this leads to microtubules that cannot break down, and therefore microtubules cannot do their job. This blocks the cell cycle at the metaphase-anaphase transition, and anaphase cannot occur, and therefore cells can no longer divide. If we go back to this slide depicting metaphase, you can think of the taxols as drugs that lock the chromosomes of the mitotic spindle in this position so that they cannot begin to separate the chromosomes, and therefore the cell cannot leave metaphase and enter anaphase. The specific target that taxols bind to is beta tubulin, and so for this reason, one of the ways that tumors can become resistant to taxols is if the tumor cells alter their beta tubulin. Another very well-described mechanism by which tumor cells become resistant to taxols is by increasing tumor cell production of P-glycoprotein. P-glycoprotein is also called the multi-drug resistance protein, and it's a protein that can pump foreign substances out of cells, including taxol drugs. And so increased production of P-glycoprotein is a mechanism by which many cancer cells become resistant to lots of different chemotherapy agents, including chemotherapy agents like taxols. These drugs are mostly used on solid tumors, including ovarian and breast cancer. They're used on both small cell and non-small cell lung cancer. They're used in head and neck cancers and also in prostate and bladder cancer. A major side effect of these drugs are hypersensitivity reactions. This can occur in up to 30% of patients. Patients can develop symptoms of a hypersensitivity or allergic reaction, including dyspnea and wheezing, urticaria, or hypotension. This is a picture of the arm of a patient with urticaria, which can occur in response to Taxol administration. For this reason, pre-medication is usually done to prevent hypersensitivity reaction. Patients are often given glucocorticoids or antihistamines in order to prevent allergic symptoms. There is a variant of the Taxols called nab-paclitaxel. It also goes by the name Abraxane. It is albumin-bound paclitaxel, and because it is bound to albumin, it has a lower risk of hypersensitivity reaction, and when this version of the Taxols is administered, pre-medication is not required. Taxols can cause myelosuppression by inhibiting the ability of bone marrow cells to divide and go through mitosis. In addition, they can also lead to neuropathy. Remember what I told you before, axons are very long cellular structures that are heavily dependent on microtubules for transport of nutrients and other substances. For this reason, drugs that interfere with microtubules can affect the nerves. In the case of Taxols, it often presents as a sensory nerve problem, usually with burning paresthesias of the hands or feet in a patient receiving a Taxol drug. The second group of microtubule inhibitors are the vinca alkaloids. Drugs in this class include vincristine and vinblastine. These drugs are derived from the periwinkle plant, which is also called Vinca rosea, and that's how they get the name vinca alkaloids. These drugs bind to beta-tubulin and they inhibit polymerization. This will prevent the mitotic spindle from forming properly and the cells will be arrested in metaphase. Going back to this slide showing metaphase, you can think of the vinca alkaloids as drugs that prevent this mitotic spindle from forming properly. Therefore, the chromosomes cannot line up on the metaphase plate and the cell is arrested in metaphase. These drugs are used in a number of malignancies. They're used in breast cancer, germ cell cancers, lymphomas. They are part of the ABVD protocol that's commonly used in Hodgkin lymphoma. ABVD stands for adriamycin, bleomycin. The V is for vinblastine and the D is for dacarbazine. The vinca alkaloids can cause myelosuppression just like many chemotherapy drugs. They have rarely been associated with SIADH. And then finally, they can all cause neurotoxicity. In particular, vincristine is famous for causing neurotoxicity. This is the dose-limiting toxicity of the drug. This occurs because of loss of axonal transport because axons are heavily dependent on microtubules. This can be a sensory and motor neuropathy. It can present as paresthesias and pain in the fingers or feet. It can also present as distal weakness. It tends to affect the most distal parts of the body because those are the ones that have the longest axons that are most heavily dependent on microtubules. And that concludes our module on microtubule inhibitors.